group of fungi we're going to look at here is called the Ascomycota. And these are fungi that have septate mycelium and sexual spores that are produced internally in what they call the ascogonium, or the cell in which these spores are produced, and that would be your sexual spore. Now let's take a look at the way this works in the life cycle. It says we start out with the septate mycelium, and if we start on the left hand side up on the top, you see cells that are what we would call dikaryotic, meaning they have two haploid nuclei on the inside of. These dikaryotic cells in this specialized process begin to turn around and bend around, and what you get is you get division, where you create two cells. One cell will have one of each nucleus in it. The other ones will have a single, they will be haploid. The two nuclei come together, they fuse, you get meiosis followed by mitosis, or mitosis followed by meiosis, in which case you end up with 8 haploid spores. These haploid spores, by the time we get over to the right-hand side, form little cell walls around the outside of them. And as we do that, we now have 8 new sexually formed spores, because they went through this process of meiosis, and these are 8 haploid spores. So out of this, you get this process occurring. Now, this occurs in a layer that's often called the hymenium, and on this particular structure here, you can see the fuzzy little layer up on the top of it in a line-like format. You can see how it looks like a bunch of columns. If we look at it much closer, what we see is this. And in here you can see spores, and if you count them, you'll find that they are in groups of 8. They are inside of these cells, so the cell would be an ascus, and these would be called ascospores. These spores are haploid. Now, when we see these things, they can produce a tremendous number of spores in these, and these spores are then disseminated so they can propagate the fungal material from there. If we look at reproductive structures, these asci can be formed individually on the outside of things like plant tissue, or in the case of a yeast, what we've got single-celled, and in this case, we would call these a naked ascus. We can see in this particular picture, which is Taphrina deformans, this is something that grows on peach leaves. And if you look at this, you can see where it's being colonized on the outside. Then up on the top, what you have is the production of these asci with the ascospores on the inside of it. So in this particular case, you're looking at what we would call a naked ascus. Here we have a cleistothecium. A cleistothecium has the asci scattered on the inside. It is closed, circular in shape, and the spores are released after it breaks open, and that would be represented by this. And you can see the asci on the inside where the spores are being formed. We could have a perithecium. A perithecium is flask-shaped. You can see the opening on the top. The opening is called an ostiole. It's a little pore out of which things come. And if we look at that in a photographic manner, we see something like this. And you can see the asci down at the bottom, and they all kind of point towards the opening in the flask. We have what we call an apothecium. An apothecium is open at maturity. And you can see the layer in here of asci and ascospores, and that would be represented by something like this. This is on a leaf, and what you can see in here is this structure is opened up, and that kind of creamy area on the inside is full of asci and full of ascospores. Or we could have what we call an ascocarp. An ascocarp is where the fungal tissue comes together and it kind of forms an amorphous mass. And on the inside of these amorphous masses, we have the formation of the asci. And basically what you get is you get a little chamber, and the asci get formed on the inside of that. And that would be represented by something like this. This is really a mass of fungal tissue, and you can see the little things that almost look like little perithecia with little openings on the inside of each one of these structures, and each massive structure is called an ascocarp. So we've got 5 different kinds of reproductive structures that we deal with. When we deal with yeast, we talked about these. We said they normally produce naked asci, and what you can see on plant tissue is you see that, and that causes what they call peach leaf curl. Or you can see something like this, where you've got individual little cells. They reproduce by budding. The budding starts out where you get this tiny little cell being produced from the mother cell. The tiny little cell is called a daughter cell, and then that will separate and it will be released and it will grow from there. And out of yeast, we get things like, what is this? We got bread, we've got wine, and all of these things come from yeast. This is a cleistothecium, and you can see the asci on the inside of it. And you can see how it's really made up of different fungal tissues. This is the parathecium. This is a leaf spot, and on the inside of it, you can see various ascocarps, and these would all be producing new spores. This is an apothecium on a leaf. This particular type of leaf spot, we've got a video of this, and you can actually see these opening up. This leaf spot is caused by the fungus. Later on in the life cycle, it produces these little apothecia on the top. It gets wet, they open up. This is a grass with ascostromas on the outside of it, and you can see how the ascostromas are damaging the grass. This is another leaf, and you can see the parathecia here, and what you see is that they actually formed in rings on this particular one. This is very nasty. This is Sclerotinia sclerotiorum on Brussels sprouts, and you can see all the white mycelium growing all over, and you can see how it's destroying the tissue of the plant. And this particular plant was, never made it. We removed it and we eliminated it. And after a little bit of time, it produces these dark little masses, and these are called sclerotia. They are small, they are hard, and they will drop off of the plant. They'll stay in the soil, and then under proper conditions, they will germinate and they will produce an ascocarp out of the top of it. In this group, we also have what we call the mitosporic fungi. Mito for mitosis, sporic because the spores are produced through mitosis, which means they're not sexual spores. We often call these types of spores conidia. And there are a lot of different ways they can be formed and a lot of different things that we can see on them. In this particular case, you can see septate mycelium coming down. They branch off and they produce these little cells, and on the end of the little cells, you get the formation of the spores. These little spores are kind of oval shaped, and they are called conidia in this particular case. This is penicillium. This is what it looks like when it's growing on culture media or other things like that. And you've probably seen this growing on an orange. You've probably seen it growing in blue cheese or on brie. Other types of fungi can look like this, and you can see the mycelium. This was isolated out of leaf tissue. If we take that and we put it on a slide, you can see the septate mycelium. You can see they come up, then they've got these little platforms on which the spores are produced, and the spores in this case are called conidia. And these are quite large compared to things like the penicillium type. This is aspergillus, and in this particular case you can see the elongate conidiophore. That is a structure that produces the conidiogenic cells on top of it, and then you've got this little head, and the little head is producing these spores, and you can see the little dots of spores all over the place, and it produces thousands. So these are called mitosporic fungi. On plants, they can do things like this. This is crepe myrtle. The little white dusting on it is all fungal spores. On an orange, you've seen that. It's nasty, but it's all covered with fungal spores. On an apple, this is penicillium on an apple. It starts out as this little thing. It creates this sunken depression on the inside of it, this wet rot, and that's caused by a mitosporic fungus. You cut it open, you can see the wet rot moving down in, and up in the center of it, you can see where it's starting to produce spores. And if you let it evolve over time, it will produce something that looks kind of like this, which is really nasty. This is banana, and the peach-colored material on it is fungal spores. Now, this particular infection occurs in the field when the banana is just starting to form, but it never goes anywhere until the banana starts to get ripe. All the little dots on the outside of the banana are caused by this particular fungus, and you only see them occur when the banana starts to ripen. If you eat it before it gets to this stage, you'll never even know it was there. But it's absolutely harmless. Now, I'm not going to say we're going to eat rotten looking banana or anything. So what you do is you cut this part off and the rest of it's fine. This is a fungus on the roots of a bean plant, and you can see how it's destroying that particular plant. So when we deal with the Ascomycota, we're looking at fungi that have septate mycelium, but when they produce sexual spores, it's on the inside of cells, and they often produce what we call conidia, which are the asexual spores, and the asexual spores, we often call those particular types of fungi mitosporic fungi.